Hello, friends. In this uh, lecture, I am trying to introduce the subject of experimental design. You know that there are many types of designs: experimental, descriptive, uh, grounded theory approach, qualitative. Uh, it depends on your requirements, on uh, and also on the literature available, your assessment of literature, what kind of methods have been found to be more suited to study a particular problem. Experiment, in short, I can say before I go to my slides that experimental design is used when you have already developed scientific hypotheses which are testable and which can be empirically falsified and you are looking for a causal connection. Causal means that there is a cause, uh, and any variation, any change in cause would eventually, after some time is not decided how much time, but after some time, it will produce a corresponding change in the dependent variable. Okay. So, now, a simple thing would be to compare two groups in which in two groups. In one of the groups, you make the experiment. In another group, you do not make the experiment. Measure their values of dependent variable before conducting the experiment and after conducting the experiment in one group. See if the values taken second time differ between control group and experimental group. If there is a difference, or if the difference after experimentation is higher or lower than the difference that existed before performing the experiment, that can be attributed to your cause. But there are complex questions associated with experimental design, and I would try to address some of them. Now I come to my slides. Uh, the title of the presentation, as I said, is experimental design. And uh, in experimental design, uh, I start with an example. How do you study effects of physical activity on children's executive function? Recently, I read a paper on this topic in which experimental method was used. So I thought that I can start this lecture with this example. Now, common sense suggests that compare two groups, those who are physically active and those who are not physically active. But why not three groups? These are the questions of research design. Why not three groups? Very active, somewhat active, and not active at all. That also can be done. That is another kind of research design. But the relationship may be non-linear, and three or more groups may be made on the basis of severity of activity, and in some of the groups at some stage the cause may be removed, means those who are subjected to physical activity, they may be withdrawn from physically active group, uh, 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 or the nature of exercise may be changed, or severity of exercise may be changed. It may be raised, it may be lowered. These are questions for designing a study. Now, common sense has obviously no answer for why should relationship exist. You have to read literature. Why compare? Why not motivate all students to take up physical exercises and observe? Then what type of physical activity? Simple, habitual, or complex? And what is executive function? A psychologist may define it as a cognitive process necessary for goal directed cognition and behavior. But it makes no sense to common people. So, whatever subject, whatever research topic you are studying, you only understand the jargon, technical terms used in study of that subject, and they must be well defined first. And this definition of your uh, variables comes from reading of literature. It will not come from sky. It is not speculative. You have to justify. As a matter of fact, in research, I used to say that there is no right or wrong answer. A right answer is one which can be justified by you on the basis of literature. A wrong answer is one which cannot be justified on the basis of literature. Only you are speculating or which is commonsensical. So compare two groups or more. It is also important to think about cost of comparison. This cost of comparison will determine how many people can be selected for comparison, how many of them can be put in the control group, how many in the intervention group, and what is the ethical aspects of the problem. You are not permitted to study all kinds of problems in social sciences or management. There are ethical aspects of the problem. Should that problem be studied at all? Or will the findings of your study lead to some undesirable consequences for certain groups or for society as a whole? Uh, for example, here a question can be asked, is it ethical to keep some adolescents in the control group? Uh, if you if you know on the basis of literature or your own experiences that physical activity is important, then why deprive some people from physical activity and lower their executive function? Now, also remember that for certain issues like executive function, you have to read literature from psychology journal, develop. This is an interdisciplinary problem. Developmental psychology, kinesiology, cognitive neuroscience, and biophysiology. Psychologists may also not agree on the same definition. So you have to make a careful decision regarding which definition to use for cognitive for executive function. It may be defined as cognitive demands inherent in a structure of goal-directed and engaging exercise, uh, and cognitive engagement required to execute complex motor movement. If you are not working on this problem, executive function, then this definition is not important for you. But this shows that whatever you are studying, you must have a good definition which can be justified on the basis of literature or about which there is consensus in the scientific community, in the community of psychologists or management experts in your field of inquiry. So when to use experimental design? There are so many designs, as I said, but this is one specific design. This design is used when you want to establish causality. Descriptive designs are good for something else. Qualitative studies are good for something else. For for understanding the experiences of people, the qualitative study is good, but it cannot establish causal connection. For causal connection, grounded theory approach is also not good. For causal connection, maybe triangulation is also not a very good method. Only experimental design, experimentally, you cannot take liberty with experimental method and do some part by using experimental method and some part by using descriptive or grounded theory or qualitative research and say I have used triangulation and therefore it is better. No, if you are doing experimental research, I would say do experimental research carefully. Read literature, identify your dependent and independent variables clearly, and develop the conceptual framework which shows the nature of relationship between your variables, which identifies proximate variables or intermediate variables or mediating variables, and also moderating variables and covariates. That is very important. Also, this experimental design is of special importance when independent variables are numeric, they can be measured, and the dependent variable is qualitative or categorical only. It can only be divided into categories and it cannot be measured as such. Now, experimental design is good for internally and externally valid conclusions. I will come to this point of validity in a moment. So, Now, it must be clear what is experimental research. It is a systematic and scientifically acceptable way of understanding the cause and effect relationship. Used mostly in lab-based experimental psychology, in agriculture, in medical sciences, in biomedical models, so that importance or efficacy uh, uh, or the working of some drug uh, can be tested in cases of certain disease. So a new drug is ready. It is ready, and its chemical and uh, biophysiological properties are understood, but it has to be tested. First it is tested on animals, and then there are clinical trials. These clinical trials require controlled experimental method. As a matter of fact, Baba Ramdev has made this controlled experimental method a household term. Everybody understands what this is compared to groups. The simplest form of experimental design is the randomized controlled design. I will explain what randomized means. Compare two groups. In the above example, one subjected to habitual exercises for several weeks and another control group which is not subjected to exercise. This is the ideal form of randomized control trial. So you have a group called intervention group, another group called control group, and you have a population from which your subjects or respondents or patients, uh, they can be selected, your participants. This is the universe of participants. And you use some method of selecting some of them, means you do sampling. And uh, one of the method of sampling, which is often desirable, is the technique of randomization. Determine your sample size, and uh, um, after determining sample size for intervention and control group, you select some participants randomly. Randomly means by uh, ensuring that the probability of selection of each of these persons in the intervention group or in the control group is same. So select randomly. Why randomly? Because uh, randomization ensures that with respect to other characteristics which might influence the outcome, or which might help in the treatment of the disease, uh, uh, if sample size is sufficiently large, they are standardized. That both intervention and control groups will have the same composition. For example, if you think that the effect of the drug uh, uh, depends on gender, okay, so either you take uh, equal number of males and females in both intervention and control groups, or randomization means that if sample size is sufficiently large, what is large cannot be determined in advance, just follow the convention. If it is sufficiently large, in statistics books, it is said that a sample size of 30 is sufficiently large. So if sample size is sufficiently large, we assume that with respect to other characteristics which might affect the outcome, there are no differences between participants included in intervention group and participants included in the control group. There are several issues with this simple design, and in experimental case, Solomon four-group design suggested by Richard Solomon in 1949 is considered to be the ideal design. That takes care of history, measurement, effect. These are some sources of or threats to validity, to which I will come in another slide. What are these things? I will explain. In Solomon four group design, you have participants who are randomly assigned, and there are not two but four groups: group one, group two, group three, group four. You see, group 1 and group 3 are treatment groups in which intervention is made. And group 2 and group 4 are control groups in which intervention are not made. Now, in all the groups, uh, measurements are taken after the experiment or intervention is over. But before intervention, measurements are taken in group 1 and group 3 only. By making permutation and combinations of these measurements, now it is possible to tell a number of things. Uh, you must ensure that the assignment of participants is done at random. The outcome of interest is measured simultaneously across groups. The design is asymmetric in the sense that only participants in group 1 and 2 receive pre-test measurements. However, all participants receive post-test measurements. Uh, internal validity means, first, the simple definition of internal validity: the confidence that changes in dependent variable are actually caused by independent variable and the relationship is not pseudo. Means it does not arise because there are differences in uh, participants between control and experimental groups. It internal validity also depends on how accurate or how unbiased or how noise free your measurements are. What is the validity of your measurements? There are several ways in psychometry to determine uh, validity and reliability of measurements, and you can look into that. Can you measure them like time, speed? Perfect measurement. Sciences have perfect measurements. You can perfectly measure time, speed, mass, electric and magnetic charge. Actually, science has advanced so much, and there is so much of technological advancement before us, partly or fully because scientists have been able to measure their variables perfectly, time, speed. Nobody asks what is the reliability or validity of your measurement of speed or distance or mass or electric charge. Now, threats to internal validity normally are when you take repeated measurements. You take a measurement before conducting the study and after conducting the study, or you take measurements uh, 3 or 4 times. One is history. History means that with change of time, characteristics of participants themselves are changing. You know, and uh, jokingly, it is said that if you are suffering from infection, then it will be cured in a week's time, whether you take antibiotics or not. So, this change of history, that history, uh, passing of time, a week's time, has cured you. It's not antibiotic. In majority of cases, antibiotics serve no purpose. Infection or viral diseases get cured by themselves. This is history. But if you compare one group who is, of people who are given antibiotic and another group of people who are not given antibiotic and compare the results will be similar. So, and you will attribute them to antibiotics. But actually, this result is not of giving antibiotic, this result is of history. Similarly, maturation. Maturation means the participants, as time passes, they change their characteristics. As age increases, uh, our perceptions, our creativity, our mental stability, our mental models, our mental complexity, everything changes. So sometimes you attribute your experimental findings to independent variables, but actually they may show the maturity. Similarly, testing. Testing means when you take measurement 3, 4 times, then if the instrument is not same, or uh, there is some kind of learning between participants and instrument, then also results will be affected. Instrumentation, change of testing instruments. Suppose a doctor takes your blood pressure by using one blood pressure machine today and another blood pressure machine after 4 or 5 days and sees the change. Actually, that change may not be because of change of your blood pressure. It may be because of change of machine. Similarly, confounding factors. There may be other factors which affect or covariates which affect the outcome variable. Statistical regression is the property that uh, if if you take measurements several times and they involve people's response, that people have a tendency to move towards a middle value. Then selection bias of participants essentially, but it can also include selection bias of measurements or outcome measurements and attrition or experimental mortality. If your experiment is long, some of the respondents may be tired, some of them maybe in a few unfortunate cases they will die or they will migrate to some other place. So your values in second, third, fourth, fifth experiment are not coming from the same set of subjects or participants. The term external validity is used for generalizability, representativeness of sample, setting procedure. Uh, if you have not done experimental research, but you are familiar with survey research, I can tell you that external validity is a kind of sampling error. Sampling and survey research include sampling error. They include relative errors, confidence interval. Therefore, we have to calculate confidence interval, 95% confidence, 99% confidence interval. Uh, and they all these errors or these sampling fluctuations depend on many things, including your design, size, and estimator. These things as such do not apply to experimental design, but in experimental design also you take a sample. Otherwise, I would not say randomized control design. You may use some other strategy to select your participants. Then it will not be simple random design. And number of participants in control and experimental groups, and the estimator that you choose, mean, mode, median, these are estimators, or indices, uh, or constructs that you develop for measuring your dependent and independent variables. Your uh, external validity depends on those things also. And now, in case of Solomon research design, many researchers think that Solomon design wastes resources. It is too complicated, especially if there are multiple treatment or experimental groups, or we work with limited time and other resources. If you are a PhD student, you have a limited time. You have to submit your PhD thesis in 4 years' time. While Solomon's four-group design requires a larger sample and uh, it's a, it involves more cost. It will take more time because you are studying four groups. If you are single-handedly studying a complex design, then you will have to spend more time and resources. We are not looking for an absolute or theoretical truths. We need feasible and practical answers. So if we need physical, feasible and practical answers, maybe simple designs are better. They are as good as complex design. The statistical theory of Solomon design would be too complicated. We can understand simple ANOVA, but not beyond that. I will explain ANOVA. We need simpler designs, easy to understand, easy to use, and easy to communicate to planners, donors, managers, social action groups. As I said, that Baba Ramdev has popularized this term, randomized control trials, for showing that his medicines, his buttis, his churn, his asavs, uh, his grids, madhu grid, orthogrid, and all these medicines are based on randomized control trial. And his doctor, Varshney, explains before the public, before the audience, that two groups were selected. two groups of mice were selected. In one group, they first infected them with some disease, uh, and then they became part of intervention group uh, and control group, and some drug like orthogrit or madhugrit or some bati would be tested on them, and if it is found that intervention group is doing better, huh? You understand that first all the participants or all the mice included in both control and experimental groups are infected with certain diseases, some maybe say cancer cell, and then a cancer medicine is given to intervention group, not to control group, and results are compared. If the drug or if the medicine is effective, then those in the intervention group will be cured. And only after establishing the findings of these kinds, the drug will go for human trial. Uh, so sometime for simplicity and PhD type research, we do not use experimental, control experimental, or randomization may not be possible. So we go for matching groups. That becomes a quasi experimental or non-experimental design. I cannot think, you may have a feeling that I cannot think of applying a randomized control trial, or where randomization also requires a complete listing of all the participants before you randomly allocate them to control and experimental groups. Okay, then I will go for a quasi-experimental design. It is as convincing a design as experimental, but it is not experimental. Uh, it is a non-random design or based on multiple measurements. Single subject experimental designs are also possible, and they are called quasi experiment. Single subject means you take only one participant, uh, give a drug, see its effect. You must have heard that homeopathic medicines were developed like this. That a normal person was taken, and some drug uh, was given. Some medicine was given, like Bryonia 30 may be given. And if this normal person becomes sick, then and develops certain symptoms, then it is believed that Bryonia Bryonia will cure all those persons who have similar disease symptoms. This is single subject experimental design. There is an experiment. What is experiment? The normal persons are selected, and some specific drug, arsenic or bryonia or belladonna, is given, and it is examined. What symptom do they develop? They must become sick, because this is not part of their daily diet. This is not normal to take arsenic. So arsenic in potentialized form is given to a single subject. Then symptoms after giving the drug are noted, and it is claimed that all those people who are suffering from similar symptoms will require this particular medicine. Similarly, non-experimental qualitative case study. Sometime with single person, we take repeated measurements as shown in this slide. Repeated observation, observation, observation. Then an experiment is done. Again, observation, observation, observation. Then the experiment is withdrawn, and then again measurements are made, observation, observation, observation. Then again experiment is done or intervention is made, X, and again observations are. So there is only one single case. As far as number of participants is concerned, there is only one participant. But on the basis of multiple measurements of this type, we can study the effect of X or intervention or the independent variable. The advantage of this kind of design is that we can also see, I raised a question originally, uh, whether uh, uh, whether improvement in physical activity leads to improvement in uh, executive function, and the relationship is linear. If relationship is linear, every improvement in physical activity would lead to a corresponding improvement in executive function. But this may not be so. After reaching a certain level of after a level of severity of activity, there may be no further change in executive function, or the executive function may start uh, declining. So, this kind of reversal time series design with one participant may answer that kind of question. Now, how to take care of other causal factors? There may be other factors. Uh, I call them covariates or confounding variables. Uh, you go for matching. So, in the example, when I said uh, your physical activity or the effect of some medicine may interact with gender, ensure that both uh, control and experimental groups have equal number of males and females. This is called matching. But matching means you are using the logic of uh, randomized controlled trials, but you are not using the experimental method. It is semi or quasi experimental method or non-experimental method. Non-experimental method using the logic of in experimental method, randomization is very, very important factor. Uh, you may also use a mix of randomization and mixing, because randomization leads to matching only with large sample. So if your sample is small and you go for randomization and you find that control and experimental groups differ with respect to other characteristics which might affect the findings, then you use the mixing of randomization and uh, uh, mixing. So that means matching, randomization and matching means randomization and matching means you first randomize and then uh, try to ensure by matching that uh, uh, the characteristics of the two groups before conducting the experiment become similar. The criteria for matching depend on your conceptual understanding, which is derived from review of literature, discussion with experts, attending seminars and workshops on the subject, and a note from your research supervisor if you are a PhD student. Okay, so uh, to repeat, in experimental research, you go for randomization. If it is not possible, or if sample size is small, you can ensure uh, the similarity um, of participants in the two groups through matching, and the criteria of matching should come from the reading of or from review of literature and discussion with experts. Means a good understanding of what uh, experts are saying or what has been known in this field by the previous researches. It also, whether you go for experimental, non-experimental research, also depends on is randomization or matching possible. Uh, in case of voluntary sampling means you ask people, uh, whether they are willing to take a new drug for cancer or blood pressure, which from the perspective of medical scientists are effective. New drugs which are from their point of view on the basis of animal trial they find that they are effective. Then you may ask some people to volunteer. Now, if your sample of volunteers, then randomization is not possible. And uh, you have sufficient time and resources to do multiple testing. You want scientific results based on internal and external validity or some tentative evidence for policy recommendations. So complexity of research design will depend on that. And also on what will ethical review board ask. This is another design which is practical, that you take observation. This is called uh, pre and post observations, control and experimental groups, take observation. They are not random. Uh, They are not random or but matching type, and uh, uh, you perform experiments and take the reading. In both the groups, you perform experiments and take the reading. Uh, COSI experimental, as I said, that if randomization is not resorted to and it's you are doing an experiment through matching, then it becomes COSI experimental. And time series design is certainly a COSI experimental design, as a previous slide showed. Uh, uh, Major and remember that if you are a social scientist or marketing researcher or a management researcher, scientific developments in uh, uh, time, distance, and mass. Uh, uh, Scientific developments were not possible. I have already said this, that scientific development were not possible if time, distance, and mass were not measured. Why I am repeating this? The reason is that I want to say that it is not enough to ensure that you are using causal or experimental method. It is equally important to decide how to measure your dependent, independent, confounding, intermediate, or mediating and moderating variables. Measuring concepts in social sciences is certainly challenging. How do you measure religiosity? Suppose you find you want to make a connection between religiosity and buying behavior. How will you measure religiosity? There is not one single measure of religiosity to serve your purpose. There are so many, as suggested by sociologists and all those who have studied religion. Which one to pick up? Similarly, belief in Marxism, mental disorder. How to measure mental disorder? There is no good measurement to measure mental disorder, happiness, life satisfaction, and so on. Knowledge of HIV, intention to complete PhD program. How will you suppose somebody wants? Suppose your group of experts or faculty members want to measure how good is your intention to complete PhD program. It will it will be very difficult to develop a measure for that. All measures are tentative. Teaching potential at the time of appointment of faculty or morale of industrial workers. Although there are so many measurements available to measure morale of industrial workers, but none of them is perfect. So ask yourself, are your scales to measure variable, nominal, ordinal, interval, ratio scales, they are perfect or they can be improved. So you know, although I am talking to you about experimental method, I feel it necessary to mention that these things are equally important. Review of literature and measurements of your dependent and independent variables. They are very important issues in getting good results from experimental method. How are data analyzed in experimental method? The logic is very simple. You know the idea of variance or standard deviation. You have measurements in control group and experimental group. When you take measurements, you will find that there are variations not only between groups but also within groups. So you can think of three types of variability rather. One, variability among all the participants, variability between groups, and variability within each group. And you can sum variability within each group and arrive at some kind of measure of within group variability. If the analysis of variance uses F ratio and F distribution, which has 2 degrees of freedom, one of numerator, another of denominator, and it compares variance between groups with variance within groups. There are statistical ways of fixing thresholds beyond which you can say that there are significant differences or below which there are insignificant differences. But the idea is this. If the design contains only one cause, you have one-way ANOVA. If you have two causes, or in case of physical exercise, you have three groups, uh, uh, you have uh, three groups or two causes, you have two-way analysis of variance. When you have two causes, you have two-way analysis of variance. I'm sorry, I did not mean groups. A number, one-way ANOVA can take care of multiple groups. That is not a problem. But if you have two causes simultaneously to be studied, then you have a two-way ANOVA. You require two-way ANOVA because total variability in the data has to be divided now into three things. One, variability due to first cause. Second, variability due to second cause. And third, the within. Within always remains. Now, since there are two variables, uh, two things you are comparing, so simultaneously. So variations caused by the first factor, variations caused by the second factor, and some measure of within variability in each factor and the total variability. Four things. This is called two-way ANOVA. If you want to study effects of three factors or three causes simultaneously, you will have three-way ANOVA with or without interaction. If the variables are vectors, you go for MANOVA. You take any standard statistics books, mathematical statistics books, and you can learn all these techniques from there. If you have covariates, means whose effects have to be removed, or variables which have to be standardized before comparing the intervention and control groups, then you have ANCOVA. And in case of uh, multiple measurements or matrix measurements or vectors, you have MANCOVA. Uh, I I must also tell you something about ethics. Uh, Consider studying impact of sleep deprivation. You say that I will take an intervention group in which I will deprive some people of sleep. Is it ethically okay? Or if you want to study impact of social media on addiction, is it a okay type of topic? Does sex influence short-term memory? I mean, male-female differences. One may ask ethical questions about this topic. Some people think that there are differences between men and women, uh, and these differences are biological. There are many other, or mostly the received view is that gender differences are social. They are because of different processes of socialization. And normally, these days, in the age of women empowerment, such topics are considered to be very sensitive and not to be studied at all. Uh, Forcing mental patients to take a new drug known for producing drug dependency. Uh, and normally, for drugs, for mental patient, it is said that you will take this drug for this much time only. But in many cases, it is found that the patients become drug dependent. So, are such experimentations okay? Is no harm policy not important? That your experiments should not do harm to people? Studying impact of communal hate speeches means you create communal hate speeches and then study its impact on political disharmony. Is it ethical to study? So I'm giving you some commonsensical examples to say that all types of things cannot be examined, cannot be permitted to be examined. Uh, Similarly, manipulation of results cannot be permitted. This is unethical. Uh, I know that these days it has this unethical practice has become very common, but it does not mean that this must be allowed or this is accepted. Manipulation of results cannot be permitted. And if it is ever discovered, then you may lose your PhD thesis, or you may lose your job if it is found that in your PhD thesis results were manipulated. Similarly, plagiarism, copying from somewhere, and publishing without giving credit to co-investigators. This is also unethical. So normally the rule is that if you are doing some work with your research supervisor, you become the first author because you have done the work, and supervisor under whose guidance you are working become the second author. There should not be any dispute before publication and after finishing writing the paper. If there are three, four, five uh, 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 persons to contribute a research, all of them should be made co-authors. And those people uh, who have helped your research in any way, in any small way, but they are not part of the research project, their help must also be acknowledged. Uh, Lastly, it is always recommended to conduct a pilot study. Uh, it is good to conduct a pilot study for experiencing the study at a small scale. What is pilot study? Pilot study is experiencing the study at a small scale, and to learn about estimates of variances, etc., on the basis of which you can determine your sample size to permit only a small error in the results. You can determine your effect size. Error can be 5%, 1%, or 0.1%, and how big a sample for experimental research should be there, that can be determined on the basis of pilot study. You also learn about difficulties you will be facing during experimentation, and tabulation and analytical plan. You will become more clear about tabulation and analytical plan if you have conducted a pilot study. Then after conducting the pilot study, you go for the main study, your framework, conceptual framework, tabulation plan, analytical plan, they are already ready. And you have justified your sample design and sample size on the basis of estimates of variances, etc., obtained from pilot study. So no questions will be asked about them. Your experimental findings will be accepted as valid findings. Thank you. This is what I wanted to tell you. So, friends, uh, in nutshell, what I said that there are so many research designs. There is exploratory design or qualitative research. There is descriptive design. A good example of qualitative uh, research would be that you go to uh, some areas of Jharkhand and study primitive tribes and see the impact of coal mining on them. Talk to them, live with them, observe them, and write about their experiences from their perspective. Here, their perspective or emic perspective is very important. In grounded theory approach, you write on the basis of data, but everything of research methodology is determined on the basis of your experiences in the field. Even sample size is determined who should be interviewed, how many people of a particular category should be interviewed. That is determined on the basis of your experiences in the field. Grounded theory. Descriptive, like consumption survey, employment survey, uh, even population census of India that government of India conducts every 10 years. The next census would be conducted in 2026. This is an example of descriptive. But in these studies, no hypothesis is made. In census, in NSSO, in qualitative studies, there is no hypothesis. Qualitative studies, as a matter of fact, result in some hypothesis which can be tested on the basis of scientific research or experimental research. We use experimental research to test scientific hypotheses, and the simplest form of experimental design is randomized control trial, in which we take two groups, intervention and control, and randomly assign participants to both the groups, then perform the experiment, see the difference in the intervention group. If there is a difference, that can be attributed to your experiment. But there are issues with that, issues of internal validity and external validity. And Solomon's four-group design is an improvement over randomized control trial. The main problem of randomized control trial, I should again mention, is that randomization does not mean that you have achieved similarity of those characteristics, all those characteristics which might affect your results other than intervention. So there is a method of matching, or you can go for mixture of randomization and matching. But matching makes your design a non-experimental or quasi-experimental. Experimental method, this term can be used only for uh, those uh, methods which use randomization. Okay, but there are certain gains from longitudinal study. There are certain gains from experiments on single participants, and there are many other quasi-experimental designs which you can read from your books. Uh, my purpose was only to introduce the idea of experimental design. And I think now this is clear to you. Thank you